Okay, so we covered pretty extensively what the membrane looks like, what its structure is, and specifically what membrane proteins look like. And now we're going to talk about function. And I think Eric has introduced actually this paradigm of how sequence encodes structure and structure encodes function in globular proteins. So here we're talking about similar things. And basically we're discussing how architecture of biological objects allows them to perform the role that biology needs. And we'll talk specifically about the movement of substances across membranes. So remember, this was number, I think, 3 and 4 on our list, and talking about this specific selective transport. So in that context, we need to distinguish basically two cases. We need to distinguish what is passive diffusion and what is active transport. The business of the word transport is not so well defined, so you might hear talking also about passive transport, but I'm going to try to keep consistent and discuss diffusion when basically a compound is being transported across the membrane along its electrochemical gradient. So as you know, due to entropy mainly considerations, if the concentration of something is high in some place and low in some other place, this tends to equilibrate over time, and the concentration tends to go from high to low and from low to high, right? And so this can happen across membranes. So this is what this says here. You can see these letters all say A, B, and C, high concentration, shown by these large letters, and the flux is going towards the small letters, which tend to show the lower concentrations. And on the microscopic level, you might have transport or diffusion that is going in the opposite direction, because microscopy is stochastic like this. So when we talk about the flux across the membrane, we're talking about the net flux, which means influx minus efflux. So it doesn't mean that no molecule at all goes in the wrong direction. It just means that on average, overall, the molecules are going down their electrochemical gradient. Here we have, on the other hand, this last column is active transport. And as you can see, this molecule D has been transported against its electrochemical gradient. So this is not favorable thermodynamically. And the only reason it can happen is that there's an external source of energy that has been introduced. In that case, they are showing you ATP being hydrolyzed into ADP and PI. But there can be other sources of energy, which we will discuss today. And then, as you can see here, two different classes, passive and active, I just discussed. But also, you have these types of transport that are called non-mediated, or these classes of transport that are called transporter-mediated. Non-mediated means that either the compound is able to go via the membrane directly, and this can be the case of a molecule like oxygen or dioxygen, because it's relatively hydrophobic, so it can go through the membrane directly without the intervention of any membrane protein. On the other hand, ions will be transported through dedicated pores called ion channels, but it can also be other types of molecules. So non-mediated, don't be fooled. Non-mediated doesn't mean necessarily that there's no protein involved. It just means that it's either a channel or no membrane protein, and there's no change that happens within the protein. It's just, in the case of this one, it's just a hole, basically, and things can go through. I'm going to go through examples of this if this seems mysterious. These two are transporter-mediated, which means that there will be a change in the protein that allows this movement to happen, one or the other. I'm going to give you examples. So this was glucose transport, and in that case, it's sodium being transported against potassium coupled to energy consumption in the form of hydrolysis of ATP and ADP. And we're going to talk about these in a lot more details. Here, I just want to highlight the classification. Okay, so diffusion I've just mentioned. It's a spontaneous process that tends to equilibrate concentrations over space. And the equation that describes the energetics of this is this equation. And in that case, this delta G here is the free energy of transport of this compound into the cell. Okay, so if you take a concentration here, so this would be concentration inside, concentration outside. That's what this I and O are for. If you have a concentration inside that is higher than the concentration outside, then this ratio is more than 1. So then log of this ratio is more than 0. So this whole thing is positive. So the free energy is positive. Positive free energy means unfavorable to transport the compound into the cell. And it makes sense, right? Because the concentration inside was higher than the concentration outside. Okay? So quite simple. If you do the math here, maybe I'll just write this down on the board. Actually, it's helpful, I think. So if you just take this RT and transform the log into a log 10, then you find this relationship. (1.4 kilocalorie per mole) log in base 10 Ci over Co. So if you have a difference in concentration of 10, right, you're storing 1.4 kcal per mole. If it's 100 difference, you have 140 kcals per mole, just to give you an idea of the types of energy that we're talking about. Yes? This is the same as this, except I transformed it into units where we can think directly about it. So just because this is in base 10, then we can just think about a ratio 10 to 1. And this is directly in kcals per mole, which is the unit I'm used to reasoning in. I don't know about you. I mean, it's kind of the old-fashioned unit. Some people think better in terms of kjoules per mole, but 1.4 kcal per mole gives people who are used to it an idea of what type of energy this is. So for an uncharged solute, concentration difference of 10 gives you 1.4 kcals per mole, one way or the other. And then if it's a charged species, then the free energy is not only dependent on the concentration difference, but also on the charge of the solute by this equation, where Z is the valence of the ion, F is the Faraday constant, and delta EM is the electromotive force in volts, for example. And I don't know if you guys know this, but the resting potential, membrane potential in cells is about 70 millivolts. So if we calculate the same thing, but for a charged solute, then we get, so let's say it's a sodium ion, for example, then you'll get 1.4 kcal per mole times log of Ci over Co plus Z times F times delta EM, which if you, this is the valence. So the valence of a sodium ion is 1. F in kcal per volts per mole is 23.06. Times 0.070. I just, this is in volts, because like I said, it's minus 70 millivolts. And if you just apply this, you'll see that you'll end up with minus 3.1 kcal per mole. So to summarize, if I have a concentration of glucose that is 10 times higher inside than outside, I'm storing 1.4 kcal per mole energy. But if it's a mole of sodium, I'm storing about double the energy due to the electric force. So the electric force due to this membrane potential of minus 70 millivolts kind of doubles the energy that can be stored by an electric species. Okay. Yes?
Where is the minus coming from?
Which minus? So this minus, this depends if you it depends if you consider, I think I made a mess, so here, so it always depends if you have Ci over Co and if delta G is taking if you're measuring the energy that it takes to go in or out. So here in that case, I was saying 10 times more sodium inside than outside, so it would mean a negative energy, and then the negative from the membrane potential is such that it's favorable to store sodium. It's a question. I'm making just qualitative arguments to give you. I'm making quantitative arguments to give you an idea of the balance of the energy where it comes from. So this is one thing. So this is without even talking about the membrane. Just imagine you have a structure that prevents a solution from going from one side to the other. Diffusion coupled to electric transport will make you want to equilibrate, but then you have the membrane in between. And so if the solute is able to cross the membrane, then this will equilibrate. But if the solute is not able to cross the membrane, then it will not pass. And so this is what it says here. The substance must be able to pass the hydrophobic membrane bilayer. And these are different compounds. And basically, what you can see is that the permeability in a specific tissue is directly correlated to the partition coefficient of this compound between water and octanol. So the more hydrophobic a compound is, the less likely it is to pass the membrane. It has to be it has to be hydrophobic enough to be able to pass the membrane. Yes? So there's kind of two arguments with, here is the thermodynamic force that governs the equilibrium, and this is the barrier that you have to come over to be able to transport. So water, for example, is a very important species that needs to be transported. It's not favorable to transport water through a membrane, but there are membrane proteins that support this. So this is called aquaporin. If you remember the picture where I was showing you that the lipids are tightly associated with a membrane protein was actually aquaporin. So it's 4 subunits. Here we're not showing the one that is in the front, but it's 4 repeats of the same kind. And one of them you can see here as green. You can see 1, 2, 3, 4, 5 transmembrane helices. And water is here in the middle being transported from one side to the other. So this is your maybe first example of the details of transport protein that transports water. And I don't have time to explain this here, but there was, for a long time, a question how it's possible that water is transported without transporting protons, because you might know that protons tend to jump across water. And it was resolved by which kind of structural details it was possible to transport water without transporting protons in this protein. So that was water. Now I'm going to talk about ion diffusion, and the reason I'm going to talk about ions is that they're really important for physiological function, and specifically they are the way electricity is transported in biological media. So all the excitable cells, which means neurons, muscles, heart cells, anything that needs electricity to function, is able to do so because ions get transported. So I gave you here the example of the sodium ion and how it was, its equilibrium was governed by both the electrical energy and the chemical energy. And now I'm showing you the structure through which such ions can be transported. So this is a, oh yeah, and I should mention, for biology, it's very important that there are specific channels. These are the proteins that transport ions, that transport sodium only or potassium only. So it's important that they're able to select one or the other, because that's the way then the electrical cells will be able to function properly. And this is the structure of the first potassium selective channel that was resolved in 1998. And the author of this got the Nobel Prize for it. And it's made of 2 transmembrane helices that you can see here. So the membrane is here, right? You can imagine that you have the hydrophilic head groups here and the hydrophobic tails here in the middle, like we've seen before. So you have 2 helices on each side. And here you can see 2 times 2, but in fact it's a tetramer, so there's 4 times 2. You're just not seeing the 2 helices that are in the front and the 2 helices that are in the back. But it's a tetramer, and the potassium ions can go through this axis from the intracellular medium via this gate that is actually open if you look at it in 3D, so it looks closed here, and then through the selectivity filter. And so these channels select specifically for potassium, and the channels that transport sodium look completely differently. And it was, for a long time, people wondered how it was possible to select for potassium ions, because potassium ions are smaller than sodium. So it's kind of easy to imagine how you select for something smaller than bigger, but how do you select for something bigger rather than smaller? And now we kind of understand how this is done. And I'm going to show you a movie of what happens in this region when the ions are being transported as a result from molecular dynamics simulations. And you can see the potassium ions, those are the big spheres. When an ion comes in, it kind of just pushes the other ions further. So this is called the knock-on mechanism. You need an ion to come in for the next one to be pushed out. And because of the shape of the protein here, a sodium ion is actually too small and too hydrated to be able to go through. That's how it selects for potassium. I don't know if this looks impressive to you, but it was a mystery, for decades, and people were very excited to be able to explain how this happens. Other than being selective, like I was just describing in this region that is called the selectivity filter, the channels also need to be able to open and close, because as you can imagine, you want the cell under some conditions to be able to conduct and under some other conditions to not conduct. So gating means opening and closing, or closing and opening. And different channels will be gated by different stimuli. Stimuli means the external thing that changes the properties of the channel. So this one I just showed you from bacteria. It's gated by pH. If you change the pH, you open the gate or you close it. But more advanced families of channels are gated by other things. So some of them are gated by changes in the voltage or the membrane potential. I just told you that the resting membrane potential is minus 70 millivolts, but when there's an electrical signal, like an action potential that comes, this changes the voltage, and these specific channels, they're in charge of conducting ions in a voltage-dependent way. There are channels that are gated by ligands binding, and there are channels that are gated by mechanical types of action, and we're going to just see a couple examples of those. So this is a voltage-gated channel, and it's basically made of the same pore that I just showed you before, plus many kind of decorations around it that will make it voltage dependent. So you recognize maybe this part here. You recognize that it's a tetramer again, so 4 subunits. And you can see here the 2 helices that I showed you before that were making one of the subunits. And this has also another domain here that is in the membrane. So the membrane is here, right? And then it has intracellular domains that are mainly important for the channel to come together as a tetramer. But I want you to focus on this specific region, which is called the voltage-sensitive domain. It's made of 4 helices. And this S4 helix, if you look at its sequence in terms of amino acids, you'll see that it has a lot of hydrophobic residues in red, but also a lot of positively charged residues, arginines, lysines. And remember what I told you in the first part of the lecture, we don't want to have positively charged residues in a transmembrane position because they're hydrophilic, so it's thermodynamically unfavorable. What can we say if nature has put a lot of unfavorable things in a place? Is that mysterious for you? It's okay. So, but if it's unstable, evolution should have gotten rid of it, right? So if nature has, up to this point, in a very advanced life form, kept these positive charges, it must mean that they're crucial for function, right? Does that make sense? This is kind of a central thing that you need to think about anytime you bring up biological arguments. And I just said that these things will be in charge of sensing voltage, which is important for electrical signals, which is important for brain function, muscle function. So you can imagine in animals, it's really important. And so the way it works is if there's a change in the membrane voltage, which means the difference in voltage between the 2 sides of the membrane, then imagine all these positive charges. They can sense the change in the voltage, and they can move in response. Yeah? So this is a very kind of cool device, if you want, that can sense changes in voltage, and that can then respond to it. And through some mechanism, then it tells the pore that it has to open or close, depending on the membrane potential. So I have a movie to give you an idea of how this looks like. And again, this is molecular dynamic simulations. So the helix that has all the positive charges is this red one up here. And the positive charges are shown here as sticks. And then this is the pore region, right? And I'm showing one subunit from the side, but it's actually a tetramer. But I'm only showing you one subunit. So look at that. The potential is oriented in this way. You can see how the red helix is being pushed down. And this is more difficult to see, but basically it's causing the pore to close as the helix is going down. I'll play that again without talking. So overall, the helix is moving down. This is made possible because you have all these positive charges. And one thing I want you to kind of see is also how dynamic this is, how the function is dynamic. So, often you'll think about cartoons or something that just look like one state or the other. But it's a simulation, so you might argue it's not reality. But it's supposed to reflect, it's based on physical principles, right? So it's a simulation of what the biophysical object actually looks like. Okay, so with this, I wanted to show you that we started from a quite simple object, this bacterial channel. Then we attached a domain to it that made it voltage sensitive through this very specific helix. Ion channels also need to inactivate. So I'm not even going to say much about this, but just that there's another part of the channel, this intracellular domain, that can come and close the pore to make it inactivate. But I really want to talk quickly about ligand-gated channels, because they're also very important. So I think you're all aware that the way brain cells communicate to one another, neurons, is through synapses. Sounds familiar? And basically, as I described with these voltage-gated channels, the way the electrical signal, the nerve impulse, is transported along the neuron is through these voltage-gated ion channels. But then the electrical signal kind of reaches the end, and it needs to be transported to the next cell. And the way it does this is through these so-called neurotransmitters. Yeah? So you've heard about GABA and acetylcholine and that sort of thing. So these are all neurotransmitters. So basically what happens is when a nerve impulse arrives, it signals to the cell that the first cell, the presynaptic cell, that it has to release typically acetylcholine into this synaptic cleft. And then the acetylcholine will come and bind to some channels on the other side. And the binding of this ligand will cause the pore to open. And then sodium ions to come in. And the next electrical signal can go through along the next cell, because there are voltage-gated ion channels. So this is what these channels look like. They're, in a way, completely different from the other ones, but still made of transmembrane helices. So these, instead of being tetramers, they're pentamers. 1, 2, 3, 4, 5 subunits, all slightly different. And each subunit is made of 4 transmembrane helices. The one that is lining the pore is shown in blue, and the other ones are shown in red. And a quite big extracellular domain that is mostly made of beta sheets. And on this cartoon, you can see that acetylcholine will bind to the extracellular domain in these beta sheets. And this signal will be transported to here, the pore, and signal it to open and close. This is one of the most fascinating things probably still today, is this so-called allostery. So a bit similarly to the voltage sensor and how it transmits the information to the pore. It's kind of mysterious how binding of a ligand up here will then be translated into information that is available at the level of the gate. And we and other people are working really hard to understand this still. So this is one of Eric's main research directions. And in case you care, I work more with these types of channels. So we're kind of a team that combines everything together. Yes?
During an action potential or close to it, it's in another course we talked about that it was there can only be one action potential, and then there's a waiting period until the next.
Part of it is this inactivation process that makes these channels unable to reactivate directly. It's also true, so again, the voltage-gated ion channels are responsible for propagating the action potential, and these are responsible for initiating the action potentials. And these also have a desensitized mode. So somehow it's also not very well understood. When a ligand has been bound for a long time, the channels tend to close and not be able to return back to this place where they can activate. So, yeah, it's encoded in the biology in a way that we don't completely understand yet. But I think within a couple years we can tell you. Okay. Am I going very slowly? Yes. So we won't have time to talk a lot about signal transduction, so I will say a few words. But before that, I want to talk to you about this facilitated diffusion in the case of glucose. Glucose is, of course, really important because it's a source of energy, right? You eat sugar and then you have energy or something. So it's important for the cells to be able to import it. And this is done contrary to previously I was just talking about how the channels allow the ions to go through without really a conformational change. So basically it was a hole. Once the channel is open, it just transports ions all the time. Glucose is enabled by a facilitative transporter. And this means that you have the protein in a conformation where it's open to the extracellular side. Then glucose comes, binds in a specific pocket. This causes a conformational change where the transporter opens to the intracellular side. The glucose then is able to dissociate and come inside of the cell. And then the transporter is reset to this conformation where it's open to the extracellular side again. And this kind of mechanism is much slower than to transport ions, because you can imagine you have to have these conformational changes in the protein. But they can be also much more specific, because it's not a hole. Instead, it's a machine, let's say. And of course, this image that I showed you was really out of scale. So now I'm going to show you what we know about this type of transport at this scale. So look at this small molecule. This is the glucose molecule. The membrane is here again. It's not shown, but you have to imagine it. And this protein is made of 2 domains. So on this movie, this is the entire protein, actually. And you see here it's open to the outside. And glucose comes in, binds, causes this conformational change, where it opens to the inside, and then glucose can be released to the inside. And once glucose is out, it resets back to the original, to its original position. I should say this is not molecular dynamic simulations. It's just a movie that is made of different structures that were solved in different states, just kind of put together so that it gives you an idea of what it looks like. But this is why this movie looks very smooth, whereas the molecular dynamic simulations looked very shaky before. Yes?
Is it what you call active transport?
This is still called passive, because we're transporting glucose along its electrochemical, in that case, along its chemical gradient. But, thank you. We come to active transport now. And active transport, as I said before, requires an input of energy. And the input of energy can be either, remember this example I gave you before, hydrolysis of ATP, but it can come from other energy sources too. So you can couple it to a process that gives energy. We'll see an example of this. Or it could come from light. It could come from electrons, from many other sources. So examples here, the most important of all. And this is the protein that is actually responsible for creating this minus 70 millivolt transmembrane potential that is so important for physiology. It's called the sodium-potassium ATPase, or the sodium-potassium pump. And the way it works is kind of similar to what you've seen before. Let's say from here, you start with a conformation that is open to the outside. 3 sodium ions will be able to come in. Then there's a chemical reaction where a phosphate group gets attached to a specific residue. And that gives energy to the protein, which causes a conformational change, and the sodium ions are released to the outside. In that conformation, potassiums can come and bind, 2 of them. When potassiums bind, it releases these phosphate groups and causes a conformational change where the pump opens to the inside, potassium gets imported. And through this, basically, you're moving these solutes, sodium and potassium, against their gradient. So, and this will enable, so through this repetition of things, the extracellular domain will be very full of sodium. The intracellular domain will be full of potassium. And this then allows the nervous system to work. Yeah, coming in a second. You can also see that you're exporting 3 sodiums when you're importing 2 potassium. which means that you're accumulating positive charges in the outside. And this is why you have a minus 70 millivolts transmembrane potential at rest. So if you have only these pumps that work, then the membrane potential stays at minus 70 millivolts. It's a very important protein. From ATP. Where does ATP come from? So ATP, yeah? ATP, does anybody know? Yeah? Glycolysis. Exactly. So I don't know much. This is biochemistry, so this is outside of my comfort zone. What I can tell you is that ATP is the main way that energy is stored in cells, and really it comes from the hydrolysis of glucose that is then stored into these ATP molecules that can then be used by the cell. Cool. Glycolysis is, complicated cycle that you don't want to learn, but really everybody should know. Quickly, this is a bacterial protein that uses light to drive ion transport. So there's a so-called fluorophore here, retinol. When you shine light on it, it changes conformation, and this allows protons to be transported in. And this is, again, against the electrochemical gradient. So it's a way to import protons when it's not favorable. And this is important it's a light-sensitive process, so it's kind of interesting. And then I think this is my final thing I want to say on transport, going a little bit fast. This is about importing glucose, which, again, will be important for then storing ATP. And so imagine you're eating, and this would, I forgot now what this is. I think this is in the intestine, basically. So you need to absorb glucose from the food, right? And it's not very, very favorable to just accumulate glucose. So you need to couple this to something else. So these transporters, they couple sodium transport to glucose. And remember, because of that pump, we have a lot of sodium outside of the cell, right? So now what this does is that it imports two sodiums along the electrochemical gradient. And if you want, the glucose kind of piggybacks the sodium to be imported. So this is the way that we are able to absorb more glucose, even though it's already very concentrated inside of those cells. And if you solve this and consider here that you're conducting 2 sodium for 1 glucose, using this value here, that 1 mole of glucose enables to store 3.1 kcals per mole, you can solve that you can create a gradient of glucose that is 23,000 times. So meaning you can store 23,000 more glucose into the cell than outside, just because of this minus 70 millivolts of sodium and the structure of this protein. I think it's fascinating, but Another type of transport that I haven't yet mentioned is using sodium gradients, again, but now to export protons. So the previous case we've seen is you're using sodium to import glucose. This is a case where you're using the sodium gradient to export protons. And this is yet another type of structure, dimer. And this is the type of motion that this molecule undergoes. Again, here it's a morph, so it looks very smooth. Basically, you come from here, sodium binds there, you get this conformational change that you've just seen. Sodium gets released. When this is down, a proton can bind to the same residue and be exported. So it's like an elevator type of mechanism where you bring sodium in, you bring protons out. Okay. Everything is clear on this sodium transport. Don't confuse this diffusion, this passive versus active, and this mediated versus non-mediated. So try to kind of fix in your brain what are channels, what are transporters, and the scheme with the green proteins is maybe the most important thing in this part. And I will conclude here by talking briefly about signal transduction. So this is another thing that happens at the cell. And the idea here is that the cell needs to be able to respond to changes in the extracellular medium. But now we're not talking about transporting things. We're just talking about integrating signals, if you want. So these messenger molecules that are here shown as these orange spheres, basically they are signaling to the cell that it needs to do something specific. Here you have kind of 3 different types of signaling systems. This is called autocrine. So this cell releases its own messenger molecules that then signal back to itself that something needs to happen inside. This is called paracrine. So this would be some messenger molecules that are signaling to the neighboring cells to do something. And this is endocrine. And in that case, it's also called hormone signaling. What you have is that a cell releases messenger into the bloodstream. And this will then signal to a cell that is completely in a different place of the body to trigger something. So often people have heard the word hormone without knowing that this is the process we're talking about when we talk about hormones, basically. So this is the way things work very quickly. We have this messenger, this first messenger that is released by a signaling cell. And this will be recognized by specific transmembrane receptors. And these receptors will eventually cause something in the cell. So when I say something, I mean it can be that you want the cell to divide, you want the cell to die, you want to synthesize more protein, you want to signal to the cell that it needs to move, and so on and so forth. So many things that you can imagine from biology that are very complicated. And this typically happens through what are called signaling cascades. And maybe you've seen these. They look like a mess. It's like, many arrows in many directions. It's very complicated. What we're interested in here in this lecture is this initial step. And basically, as the signaling molecule, the messenger, binds to the receptor, there will be something triggered at the level of the effector protein, or it will cause a conformational change that allows another protein to bind and to then eventually lead to something biological. So the main protein involved in this are G protein coupled receptors. And the reason I'm talking about them to you is because they're the main class of drug targets in the world. So it's a very, very big family. But they all have quite a similar architecture. And this is a scheme of them. So the receptor is made by the assembly of 7 transmembrane helices. And these can bind to a G protein. And when they bind to a G protein, then the G protein will cause something downstream. I'll come back to this slide in a second. I just want to show you this scheme. I think it's more easy to understand. So this G protein coupled receptor that was made of 7 helices is now a green kind of sausage-like thing. When a ligand binds, it causes a conformational change that will allow this G protein to bind. When the G protein is bound, you get a chemical reaction that causes this G protein to separate into 2 pieces, and this specific piece will attach to an effector protein. And without telling you really much, you can imagine that there's a downstream signaling cascade that occurs. And I think what's really interesting to consider is this is a cartoon, but really it's individual amino acids that are involved in this, right? And so what happens is that when the ligand binds at the extracellular domain, you get a conformational change in the intracellular domain that causes this binding site for the G protein to bind. But really, we're talking about small differences with individual amino acids that come and bind into a pocket. And these might be familiar. You probably heard of epinephrine or serotonin. Those are two different types of ligands that activate different types of receptors, have then different physiological responses. So this is to highlight that it's a very, very big family that has major, major importance. And actually, they're very important also for chemical sensing. So most of our sense of smell and taste are mediated by G-protein coupled receptors. You can click on that link and you'll see molecular dynamic simulations of these things, just to give you an idea of how things work. But I am running out of time. So I'll just say that as all the other proteins that we talked about in this lecture, having mutations in them can cause dramatic diseases. Like, that's not so dramatic, color blindness. I guess it's a little impeding, but you know, different types of diseases and cancer. And so then the final class of protein that I wanted to mention really quickly, because it's involved in cancer, so this you might have heard about also, are receptor protein tyrosine kinases. And they also lead to a change in the cascade through ligand binding. But instead of a ligand binding to a single protein, what happens is the ligand binds to a protein that then causes a change and enables these to assemble into one single assembly and then causes downstream signals. So another way that biology can signal at the cell membrane. And of course, ligand-gated ion channels can also be seen as a similar process. So we discussed those already, but if you think of it that way, they're binding a ligand and causing a downstream signal, right? So this is to highlight that they can be put in the facilitated diffusion type of biological activity, but you can also think of them as signal transducers because they bind ligand and they cause downstream signals. Okay, sorry, the end was a little bit fast, but again, what is really crucial in this lecture is this first part. So try to understand and memorize how the membrane is made and what that means in terms of structure of the proteins, and then how this structure then mediates function. And we discussed transport, passive, active, and to some extent also signal transduction. And as always, you have here some study questions that should help you to focus on what's the important aspects. And with that, I'm done. Wow, that was fast. I think this week and the next you're going to have Burke, who will talk to you about molecular dynamic simulation. So it will be a lot less biological and a lot more about how we use thermodynamic principles to simulate the behavior of these proteins. But I hope I showed you examples of molecular dynamics and how they help us with biology, actually, because it's become really quite an important tool.